Welcome, everyone, and thank you for joining us today for this series of webinars on how electron diffraction is transforming research across the fields of materials, life sciences, and chemistry. My name is Joe Ferrara, and I will be your host today. It is my pleasure to introduce today's speaker, Dr. Jessica Birch, who is our electron diffraction application scientist based in the Woodlands. Jessica did her PhD at UCLA with Hosea Nelson and then spent 3 more years at Caltech further developing expertise in micro-ED before joining us in 2024. This is the third webinar in this series of 6. In this session, Jessica will walk you through the complete electron diffraction process, including sample preparation, data collection, and structural analysis. Jessica will also explain how integrated hardware and software in a dedicated diffractometer streamline workflows, enhancing the efficiency of single crystal structural solutions and making bulk powder analysis by ED a practical approach. Before we get to the main presentation, I want to go over some housekeeping items. If you'd like to ask a question, please do so using the Q&A button. These will be read out and answered as time allows. Any unanswered questions will be answered after the session. Where applicable, please try to include the slide number so we can more easily understand the context. We will post links and other relevant information in the chat. The chat is open, but please note that if you ask questions there, we might miss them as we will not be expecting them there and they may get lost as new messages appear. We will be recording the webinar. The recording will be available tomorrow. If you have any trouble watching the presentation live, we recommend catching up via the recording as it is unlikely we will be able to resolve technical problems during the live webinar. Lastly, if you haven't done so already, we encourage you to register for all the electron diffraction webinar series. You can do that at the link I will paste in the chat momentarily. Even if you've registered for individual episodes, you can still register for the whole series. Thank you for your patience. I will now turn the floor over to Jessica for the main event.
All right. Thank you, Joe, for the introduction, and thank you all for being here today. In this episode of our ED webinar series titled Simple Electron Diffraction Workflow from Sample Prep to Structural Solutions, I'll be walking through the experimental hands-on side of electron diffraction, showing how streamlined workflows make this technique more accessible. And it's sort of a day in the life of electron diffraction using the Synergy-ED. So let's go ahead and get started by looking at our agenda for today's talk. So the first thing that we're going to do is an overview of the technique of 3D-ED and micro-ED. After this, we're going to go through some sample preparation strategies. So we'll talk about how to pre-characterize your sample, the types of samples we can look at, as well as the hands-on aspects of preparing grids and loading them into the diffractometer. From there, we're going to discuss actual data collection and processing with the Synergy-ED, both just kind of the overall workflow, as well as recordings actually doing this in real time. After this, we will see how automation is already fitting into our workflows routinely with the Synergy-ED. And lastly, we'll end with a summary going over everything that we discussed today. That brings me to the first section, which is an overview of 3D-ED or micro-ED. And I just want to mention that this is mostly for those of you who have not seen our previous episode presented by Robert. So a lot of these slides were also covered in Robert's talk, much more in depth than I'll be presenting today. But I will be going through this just so we're all on the same page in terms of 3D-ED. But if you're interested in learning more, I would definitely encourage you to go and watch that episode presented by Robert. All right, so I would say when it comes to performing electron diffraction, one of the reasons why we're really excited about this technique is this idea of breaking the 1 micron barrier. So we know that if we are interested in analyzing single crystals, We have a range of diffractometers to look at samples from hundreds of microns in size all the way down to a micron. Now, if we're interested in doing single crystal analysis of a sample that's smaller than a micron in size, then our options become quite limited. But thankfully, electron diffraction can analyze samples that are up to 1 micron in size, meaning that X-ray and electron diffraction are complementary techniques that together allow us to study a really wide range of samples. But I would say that electron diffraction is really becoming more than just a technique that you turn to when you're growing crystals that are too small to analyze by single crystal X-ray diffraction. As we're increasing our ability to look at samples in high throughput, we've started presenting this view of looking at analyzing samples in this sort of powder grain by grain experiment. So if we think about having a powder material here, then an analysis that we would likely perform is getting a PXRD pattern. And within this PXRD pattern, we might have multiple polymorphs. We could have different phases and different compounds that are all within this single measurement of the bulk powder material. But using electron diffraction, what we can actually do with this powder is go and analyze each of those individual grains and get out information much more similar to that of a single crystal analysis. So what that allows us to do is take a mixture of powders and solve multiple structures, as we see here, where we were able to solve both the structure of an API and the excipient molecule as well. And the reason why we're able to analyze these small crystals is because we are using electrons as our radiation source. So I'll walk through just a couple of differences between electrons and X-rays. The first of those is that electrons will elastically scatter from the Coulombic potential of our material, as opposed to X-rays, which are scattering off the electron density. Electrons interact orders of magnitude more strongly with our material than X-rays do, meaning that these submicron-sized crystals are both possible, and as we're going to see in the data collection side of things, they are actually required. And another reason why we're able to look at these small crystals is because we actually have less radiation damage per unit of information that we get from the sample or elastically scattered electrons. And so it might be a bit surprising then if electron diffraction is so useful, why it's sort of lagged behind in terms of popularity when you compare it to single crystal X-ray diffraction. And the reason for that is this sort of double-edged sword of working with electrons, where this very strong interaction means that electrons are scattering within your small crystals not just once, but oftentimes in pretty complex ways, including multiple scattering events, which we call dynamical diffraction. And with dynamical diffraction, we start to lose this relationship between our intensities and the square of our structure factor, which is this so-called kinematical approximation that allows us to nicely solve crystal structures. And it wasn't until deviation in the way that we collected electron diffraction data that we started to see we could suppress this dynamical diffraction. And we actually found that we're able to apply this kinematical approximation just in the same way that we do with single crystal X-ray diffraction to solve and refine our structures. So in the more classical sense of electron diffraction, you would carefully align a crystal to a zone axis and record a diffraction pattern. And that's actually where the dynamical scattering is at its maximum. In this new data collection approach, we are continuously rotating a crystal, much in a similar way to single crystal X-ray diffraction, and we find that dynamical scattering is minimized. And so what we wind up with is that we are able to perform these kinematical structural solutions and kinematical refinements. We do have systematically high R values when you compare that to a single crystal X-ray diffraction. So here I'm showing a table of a number of structures that we've solved here at Rigaku, where the median value for a kinematically refined ED structure is somewhere around 17%, and so you'll see values like this routinely in the literature. But there are a number of publications comparing the quality of electron diffraction structures to that of X-ray structures to show that the structures that you get out are still quite good. And that brings me to the name. We refer to this technique as 3D-ED or micro-ED. And that's specifically referring to this type of technique where we're collecting our electron diffraction data with continuous rotation and shutterless data collection. And it goes by multiple names because there were a number of different groups that published this work around the year 2007. So one example is three-dimensional electron diffraction, or 3D-ED, as well as microcrystal electron diffraction, micro-ED, although there are additional names out there for this technique. These are the two most common ones, I would say, so that's generally how we will refer to this technique. And a lot of the structures that you'll see reported have been refined using that kinematical approximation, but you can also start to do dynamical refinement, where we consider the fact that these electrons are scattering more than once in our material. And the reason why we would do this, despite the fact that it can be a bit computationally intensive, is because we can improve our R values and our maps, and it allows us to assign absolute structure. So I've shown an example here of a structure of tyrosine, which we'll be going through today. And the kinematical R1 is sitting here at 15%. As we start to add in more parameters into our dynamical refinement, we can not only improve the R1 value to something that's a little bit closer to that of single crystal X-ray diffraction, but we can also differentiate between the two enantiomers using a z-score. And this technique of 3D-ED was really conducted in transmission electron microscopes that were adapted for use in 3D-ED by a handful of specialized research groups that needed both expertise in crystallography as well as electron microscopy. And that's where I want to bring up this point of crystallography meeting electron microscopy. If you think about performing a single crystal diffraction experiment on an X-ray diffractometer, when we walk up to a system like this, we'll find that there's pretty specific types of measurements that you can make on this system. And these measurements are fully automated and integrated with the data that you get out. So as you're actually getting ready to set up your data collection, you already have some idea of what the intensity of your reflections will be. And this whole process is really integrated, thinking about your data in a very quantitative way. This is in contrast to performing a crystallographic experiment on a transmission electron microscope. These instruments are capable of performing a really wide range of measurements. And partially because of this means that when you walk up to an instrument like this, it's expected that you're going to have to perform extensive alignments for your particular experiment that you are performing. So oftentimes you'll spend quite a bit of time just learning how to use the instrument, learning how to align it, and configuring it for your experiment before you can even get to the point that you're going to perform your diffraction experiment. And when you do perform your diffraction experiment, the data collection and the data processing pipelines are generally separate processes, meaning that when you're actually using the instrument, what you're looking at is qualitative and mostly just image analysis visually. And you don't have this quantitative data-driven approach to your data collection. And so when you're performing a crystallographic experiment, there's certainly differences in expectations when it comes to instrumentation and analysis when you're performing a experiment on a more traditional diffractometer like we see with X-rays as opposed to electrons. And that's really the big motivation behind the development of the Synergy-ED which is a collaboration between Rigaku and JOL. And this is a dedicated electron diffractometer that is more catered towards non-specialists. So you really don't have to be an expert in electron microscopy to perform 3D-ED. And you can focus on the actual crystallographic experiment itself. And the reason for that is because we have this optimized hardware and software that all the workflows are catered towards performing 3D-ED, which allows us to have seamless workflows from instrument control to our structural solution. So I'll give you a brief overview of this now, but we're going to go into each of these steps a little bit more in depth throughout this talk. So the first thing that we'll be doing is screening for our crystals and locating them visually. We'll be collecting our diffraction data, solving our single crystal structure, and refining it in OLEX. And all of this is fully integrated within XtaLAB Pro on the Synergy-ED. And since the development of this diffractometer, there's been a really large number of publications of different ED structures from this system. So I would encourage you, if you're interested, to take a look at our publication library. We have over 50 peer-reviewed publications as of when this slide was made in December 2024, but there's many more in preparation and many more since even this slide was made. And with that, I think we're going to take a pause and move to our first polling question.
Thank you, Jessica. Our first polling question is, do you think the samples you work with are generally suitable for analysis by 3D-ED, micro-ED? And yes, no, some, unsure. And when we get to about 70% compliance, we'll go ahead and close the poll. So just a few more. All right, so it looks like everybody's answered. Thank you very much. Okay, I'm going to go ahead and end the poll. I'll share the results. And so it looks like 25 of 65 said yes, 4 of 65 said no, some 27 of 65 said some, and unsure was 9 of 65. Thank you, Jessica. I'll turn it back over to you.
Awesome. Yeah, so it looks like generally people think that they do have some samples that are suitable for electron diffraction, and hopefully those of you that are unsure will be able to address that in this section as we're going through our sample preparation strategies. The first thing that I'll do is just mention that electron diffraction has already been used on a really wide range of classes of materials. So these include things from porous materials to proteins and natural products. I don't have time to go into all of the different samples that have been analyzed by electron diffraction, but if you're interested in learning more about this topic, another application scientist, Amelia, gave a really great presentation on this recently, discussing different classes of samples that have been analyzed by the Synergy-ED. But I just wanted to mention in general that we are able to analyze samples that are mixtures or impure, things that are air sensitive. We can look at samples that maybe you only have nanogram amounts of your analyte, or samples that require solvation. So all those sort of trickier cases of analysis we can handle. And those oftentimes can be addressed on the sample preparation side of things. I'm going to talk more in general about air-stable samples and the more common ways that we're preparing samples for electron diffraction. So what is actually required of your sample? The first of those is going to be crystallinity. So if you're thinking about preparing your sample for ED, one thing that you might do is take your powder material and obtain a PXRD pattern. If you see something like the one above here, then this would be likely a good candidate for electron diffraction. We often recommend, if you're using copper radiation, we like to see peaks out past 30 degrees to theta. Most likely that means we'll be able to isolate some particles, and assuming those particles are single crystalline, then we can go through and solve the structure. If you see a powder pattern like something on the bottom, then most likely all of the particles that we locate are also going to be amorphous and we won't be able to solve the structure. So of course, electron diffraction is still a crystallographic technique, even though we are looking at very small crystallites. So crystallinity is still a requirement here. The other requirement is on particle size. So we want the thickness of our particles to be less than 1 micron. If you look at your powder under an optical microscope and you see something like that here on the left, these particles are too large, and so most likely you would want to try to solve the structure with single crystal XRD, or you can actually try to reduce the particle size. A really straightforward way to do this is to actually just put some powder material between two glass slides, rub those together to mill the particles down to a smaller size so that it looks like something here on the right. The particles that we're actually going to be analyzing with electron diffraction are really going to be too small to see by optical microscopy, so you really want to see a fine powder when you take a look at those on the optical microscope. Now that we know we have a crystalline material with particle sizes that are small enough, now we're getting ready to actually load it into the diffractometer, and so we need to discuss what we're putting our powder onto, which is a sample grid. So these are commercially available transmission electron microscopy grids. They are 3 millimeters in size, as I've shown here. And the grids themselves are made up of just a couple of components here. One of them is this metal grid, which gives the grid some rigidity and support. And then there is some sort of film in between these grid bars, which is where our particles are going to sit. And this film is meant to be as electron transparent as possible. And with these grids, you can purchase a variety of mesh sizes and types of metal, as well as the different types of support. I would say the most common ones are going to be 200 mesh copper grids, and frequently the thing that you'll vary would be the polymer support. So I'll go through a couple of the most common ones here. The first of which is continuous carbon, which is great for a variety of particle sizes, and you can get really thin supports with very little background, so these are oftentimes a good starting point. From here, we can look at lacy carbon. So this is an example of a crystal that I'm circling here. And this sort of spiderweb looking background is the lacy carbon support. And what's great about this support is that you can help your crystals not lay all on the same orientation on the grid, which can be helpful, as we'll see when we go through our data processing side of things. And then we also have these whole array type grids, also frequently referred to as quantifoil grids. These are useful if we want to think about freezing our sample inside of a solvent. If, for example, we have a protein sample and we want to keep it hydrated when we're collecting our data. And I'm showing here a video of what grid preparation actually looks like in practice. So the first thing that we're going to see is we're looking at this powder on a glass slide. If we needed to, we would have reduced the particle size by rubbing that between two glass slides before we get to this step. This step doesn't actually have to be performed under an optical microscope, but we're doing this just for purposes of demonstration. From here, you would grab your TEM grid, and we'll see that you carefully tap this grid into the powder, gathering the grains onto the grid. And from here, really you just want to validate that you see some of that powder stuck onto the grid so that you know you transferred some material on here. But that's actually too much powder on the grid, so you'll shake off the excess and be left with small particles that are just sort of electrostatically stuck to your TEM grid. And now what we see here is we're preparing the room temperature specimen rod. And what we're going to do is take that grid that we just prepared, and we're going to put it on the end of this sample holder. You can see there's a hole there at the end where the electrons will be transmitted through the sample rod, through the TEM grid, eventually getting to our detector. And now what we're doing is just securing a clamp over the top of the TEM grid. And at this point, we would be ready to actually insert it into the diffractometer and start to collect our data. Before we get to that, I just want to mention a couple of things about what makes a good sample grid. So there's a few different things. The first of them being that we want our crystallite sizes to be less than 1 micron. We would like to not have preferred orientation of our crystallites because we're limited in terms of the rotation that we have available to us in our goniometer. With this room temperature holder, we have a maximum of about 160 degrees that we can rotate our crystals. So we don't want them all to be laying in the same orientation so that we have a better chance of getting high completeness data. And we also want our crystallites to be spaced with a few microns in between them so that we can rotate them without it being blocked by another crystal or a grid bar. And you might be asking yourself how you're going to meet all these requirements when we did this very imprecise loading method of simply taking a grid on some tweezers, tapping it into powder, and shaking off the excess. It seems a bit imprecise for sort of meeting all of these requirements. But the reason why we're able to do this is because the grid is much larger than the actual volume that we're analyzing our sample. So I've shown here, this is our standard visual mode, which is about a magnification around 2500x. And this circle here represents 2 microns. And when you compare that to this zoomed out version of the grid, we're sitting inside of one of these grid squares, just a portion of it. And so we have just a fraction of a percent that we're looking at any one time. And so it's quite easy to actually just locate optimal regions during data collection, as opposed to worrying about a more precise loading strategy. The other thing that I'd like to mention is that when we are loading our samples into the diffractometer, we are exposing them to strong vacuum. So in some cases, what may happen is you will collect your powder pattern and see that you have a really nice PXRD pattern, but when you put it into the diffractometer and go to collect your data, you are not getting useful diffraction data. And a reason for that can be instability to the vacuum. And we have a way to mitigate this by actually cooling the samples before we expose it to vacuum. So this can prevent the loss of solvent through desolvation in a strong vacuum environment. And so you can take a sample where you see no useful diffraction data at room temperature, and simply by cooling the sample before you insert it, you can get high resolution data and solve the structure. So I'll also show you what a cryogenic sample loading actually looks like here. So this is an example of a cryo holder from the company Simple Origin. And what I'm doing here is cooling the area where I'm going to handle my sample TEM grids. And then I am inserting this cryogenic sample holder into the slot here. This cryogenic sample holder, I have pre-cooled this dewar with liquid nitrogen as well. And I turn this knob on the back of the dewar to release the sample grids into my preparation area. And you can see actually that there are 2 grid slots here because this is a multi-specimen holder. So I can actually prepare more than 1 grid at a time, which can be really useful if you want to look at multiple samples or if you want to try multiple sample preparation strategies with a single sample. You can do 2 of these at once with 1 insertion. So what I'm doing now is I am doing a cryo exchange. So I'm removing my old grids I could keep those stored under liquid nitrogen if I'd like to, for subsequent analysis. And now what I'm doing is I'm plunge freezing the grids that I'd like to analyze at cryo temperatures and placing them onto the end of the sample holder. The last thing that I'll do is clamp these grids into place, and then I can retract these grids to protect them from ice that may condense onto the end of the sample holder at cold temperatures, and I'm ready to do my sample insertion. So this is an example of sample insertion at room temperature, although if you're doing a cryo transfer, this process is exactly the same. You can get an idea of sort of the height of the system here as well. So the first thing that we're doing is putting our sample into an airlock, and we are switching on the vacuum pump. At this point, we have a roughing pump that is pulling our specimen down to a vacuum level that will allow us to subsequently fully insert it into the diffractometer. We have this visual feedback of a green light when we are at the point that we can do this insertion. And at this point, we're going to rotate the sample rod, which will open a valve and allow us to then fully insert the holder. And we are then at the point that we can start to perform our data collection. So the strategies that I've showed you with the room temperature holder and cryogenic specimen holder are certainly the most common ways that we're preparing samples day to day. But it's not an exhaustive look at the different possibilities for sample preparation. There's a lot of interesting things you can do with grid preparation, as well as different types of sample holders that we can use in the Synergy-ED. So I just wanted to quickly point out a few of those here. Some of them are, we have other multi-specimen cryo holders. We can insert up to 3 samples at one time. There are air-free holders that you could move your holder into a glove box and prepare your sample in an inert atmosphere, seal it in that inert atmosphere, and then not open it until it's already within the vacuum of the diffractometer. We also have the ability to look at samples under different liquid or gas environments. So we have here an example of a crystal that is actually within a solvent. And there's the ability to heat samples to elevated temperatures with heating holders. We have an example heating a moth crystal to elevated temperatures. And at this point, I think we are going to pause and answer any questions that you may have.
So we have a couple of questions. Let's answer one now. Does the Rigaku Synergy instrument have EDS capability?
Yes, there is the ability to add on an EDS detector. This is something that is a pretty recent add-on. We got ours installed actually this year, but it's something that you can either purchase the instrument with this, or it can be added on at a later time.
Thank you. Let's move on.
So this brings me to the next section where we'll be going through and looking at data collection and data processing on the Synergy-ED. So the first thing that I want to do is actually walk us through the workflow for manual data collection and mention just a couple of things about the actual experimental setup. So the first thing that we're going to be doing is locating crystals in visual mode. And I think it's important to note that we are both imaging our crystals and diffracting on our crystals with electrons. And this is the field of view that we are illuminating actually throughout the entire process. This field of view is limited by an illumination aperture that's sitting above our TEM grid. And creating the beam size that we'll see at the sample. This not being really to scale here, but this is the size of our electron beam as we're also collecting diffraction data. So when we're ready to collect a diffraction pattern, we will switch to diffraction mode, and a second aperture will be inserted. This is the selection aperture, and this aperture is actually below the sample. So this is what's represented by the circle, the smaller circle that we see on the screen, and where our diffraction data will actually come from. From here, we will switch to diffraction mode and get an idea of what the diffraction pattern is for that crystal that we just centered. And that moves us in this workflow to this point where we have our first decision tree, where we can decide whether our crystal diffracts well enough that we want to go on to collect another data set, or if it doesn't diffract very well, then we might want to locate a new crystal in visual mode and switch to diffraction mode and test this again. And I would say most of the time, depending on the quality of your sample, this is where you're spending the bulk of your time, is in this screening process, or just testing different crystallites to see if they diffract. After you have something that you think provides good quality diffraction, then what we'll do is in visual mode, we are going to center that crystal and collect our rotational data set. So I think this might differ from single crystal X-ray diffraction, where our data set collection is very quick. So you can collect data sets in under a minute, oftentimes up to 5 minutes. And really it's the bulk of your time is spent screening and collecting multiple data sets here. After you've collected that data set, then you're left with another decision as to whether you've collected enough data to actually solve and refine your structure. If you have, then of course you're done at this point, but if not, then you'll go back to the screening process to try and locate a new grain to collect another data set. And so now we're going to go through this in videos that I collected mostly in real time. So the first thing that we'll do when we walk up to the system after sample insertion is start XtaLAB Pro. If you've used XtaLAB Pro on any of the single crystal X-ray diffractometers, then this should look quite familiar to you. The first thing that I'm doing is actually turning on my filament, which is located in a similar place to where you would do this for X-rays. In this case, now it says ED. After a few seconds, we'll have our filament ramped up, and we will begin by actually collecting this mini map. So a mini map is a low magnification overview of our grid, and this is what's going to help us keep ourselves oriented as we're going through and doing our screening in multiple locations on a grid. And this mini map is taken at a magnification of 30x, and it's actually a single snapshot that's saved in just a few seconds so that we're able to get a full picture of the grid. And this comes back to that point I was mentioning earlier, that the regions that we actually collect data from are only a small fraction of the total available grid. So what we're able to do here is zoom in and see these different regions and get an idea of where there's more densely loaded particles, perhaps where there's larger versus smaller particles. So I can easily just click on this mini map and go to those regions. And we'll see here that this first region I go to, I'm largely seeing quite large particles. And if I find that to be the case, then oftentimes just going to a different region on the grid, I can find crystals with quite different sizes. So these being a little bit smaller and maybe something I might want to collect data on. After I've used my mini-map to get me in a region where there's suitable crystals, then I'll move on to my particle screening. All that I need to do here is once I've moved to a location where there's a crystal I want to investigate, I'll click on this diffraction button. This switches over my settings so that I can see the diffraction pattern and insert that aperture to look at this particular crystal. From there, I can quickly assess whether I want to collect data on that crystal or move on to another particle. And this is where I wanted to mention, we talk a lot about crystal thickness, but I think it's useful to see this in practice. So this is a really large crystal that I will be switching to diffraction mode on, and you can see that it almost stops the entire beam. There's very little of the beam actually being transmitted through this crystal at all. And this is what we mean when we say sample thickness, that samples need to be less than a micron in size. It can be tricky to sort of gauge this just looking at the crystal visually. So what I've done here is tilt the sample. You can keep track of that tilt here. And this 1 micron scale bar here, you can get a rough idea of seeing that this is around a micron in thickness. So this is why we say that's the upper limit of what we can look at with electron diffraction. And it might surprise you that these very small particles, things that you can sort of barely see, may actually still provide really nice diffraction. So in this case, the diffraction is a bit weak, but we can see that we have sub-angstrom resolution diffraction and something that is single crystalline, although I might need to extend my exposure time to get a good signal out of this crystal. But just to keep you oriented in terms of what types of particles to look at as you're going through and screening. So once I have found a crystal that I am ready to collect my data on, now what I'm doing is actually centering particles. And so the first thing that I've done is actually tilt my sample to 30 degrees. And now I'm adjusting the height of my sample so that as I go through and rotate the crystal, it will stay within the aperture throughout the entire process. And you'll notice a couple of things here. I'm actually collecting from the bottom portion of this crystal because this is a thinner part of the crystal. So you don't necessarily have to have a small crystal that perfectly fits within the aperture. And another thing here is that my crystal doesn't have to be perfectly centered within the aperture throughout the entire rotation series. The beam is essentially equivalent at any point within the aperture. So as long as the part of the crystal I'm looking at is within the aperture somewhere, then I know that I'll get my diffraction data. And this is not an issue like it could be if you're doing single crystal X-ray diffraction. So after this point, what I'm doing is finding the extremes of the tilt that I'm able to obtain. Even though the maximum possible tilt range is about 160 degrees, sometimes you may have other crystals or grid bars that will block you from fully tilting the sample. So this is something that when you're collecting manually, you'll test this before clicking this Start Experiment button, which is really where I set up my rotation experiment. So the first thing that I'm doing is I'm actually naming my grid and my data set name. We will be running this through AutoChem, so I am putting in the atom types. And the other thing that I'll adjust here is my exposure time and the rotation range that we just determined by tilting and centering our crystal. At this point, we have some automated settings that we could add into this with some post-collection analyses. But here I will click the Start Experiment button and we will start collecting our data. So the first thing that happens is we switch to diffraction mode and we'll first center the diffraction pattern. That's some of that flashing that you'll see on the screen. And then the beam is blanked to prevent as much radiation damage on our crystal as possible. And we're rotating to our initial starting angle. And then once the rotation begins, you'll see those images start to come in. And as the data is being collected, what you'll see here on the right is that within only a few frames, we'll start to get an idea of our unit cell. And this is where that integrated data collection and data processing becomes really useful because you may have impurities, you could have crystalline ice on your grid, and you can just look at the unit cell here and even do a CSD search at this point to determine if this is something you're interested in really collecting and solving, or whether it's an impurity and you want to move on to a different grain. You could at that point just stop the data collection and move on. In this case, this was what we wanted to collect data on, and as this progress bar is progressing, showing us that we're collecting these frames, AutoChem is taking these frames and indexing it, doing data reduction, and actually going all the way through the process of solving the structure with ShellX. And so This process will repeat as more and more data is collected. And at this point, once the actual data collection ends, if I'd like to, I could move this processing to an offline version so that I can get back to my screening. But in this case, I decided to just wait and let AutoChem run through the full process. And you can see here that we're able to get a solution of tyrosine here that from here, once this button here on the OLEX2 turns blue, then what I can do is just click this button, which this is the first time that I've interacted with the program since clicking that Start Experiment button. I can open this directly within OLEX, and now I'm left with a really great starting point that all I need to do is correct an atom assignment and do a little bit of refinement here, and I'm left with my final structure. This is for electron diffraction data, a really nice kinematically refined structure with an R1 of about 10.5%. So this full process from screening to actually getting this refined structure in this sort of best case scenario with a standard sample was about 8 minutes. And so it can be this straightforward in some cases to actually get your final structure. And at this point, we are going to take our second polling question.
Thank you, Jessica. Our second polling question is automation a routine part of the techniques you currently use for structural analysis? And you have the options of yes, no, partially, and plan to implement in the future. Looks like we have, here we go, a few more come in. Okay, I'm going to go ahead and end the poll, share the results. And you'll see that about 10% are currently using automation techniques, 52% are not using techniques, 28% are partially using automation techniques, and 10% plan to implement this in the future. Thank you, everybody. Jessica?
Awesome. Yeah. So that will bring us to our next section where we'll be discussing how we implement automation into electron diffraction to not only just streamline manual data collection, but also allow us to perform some interesting high throughput experiments. So to get into this topic, I first want to go back to this workflow that I showed earlier of manual data collection, where this first section where we describe as screening, and this part as we refer to it as our data collection, we use the name pre-experiment and experiment in this tool called queuing. And what queuing allows us to do is set up unattended data collection. So essentially how this will work is we will visually identify crystals that we are interested in currently, although there are plans to implement some image recognition for this in the future. But at current times, you would visually identify your crystals. And after you configure the pre-experiment or experiment, then this is an unattended data collection process. And when it comes to those decisions of whether you want to go back to a crystal that you've tested the diffraction and you want to collect a data set, or whether you're actually done and you're happy with your final structural solution, those decisions are streamlined as much as possible with the results viewer. So we sort of use these two in tandem to allow for collection of large volumes of data in an automated way with queuing. And finally, we can streamline our decisions about data quality, particularly after we run through automated processing like AutoChem, and look at that on our results viewer. So we'll walk through this process now. So this is going to be an example of particle screening or pre-experiments with the queuing tool. So the first thing that I'll do is actually center the particle that I'm interested in within this aperture and click the Add Pre button. The way that I'm going to change this relative to our manual data collection is I'm going to set a very small tilt range here. In this case, I just did 2 degrees, and I'm setting my scan width, in this case, to 2 degrees. These are just my preferred settings so that I get a single snapshot from my pre-experiment. Once I have set up my first pre-experiment, from here I can just go and identify more crystals, click Add Pre and OK, and it will apply the same experiment settings that I set up for the first sample, although if you'd like to customize these to each crystal, you certainly can. And at this point, we will go ahead and start the experiment. One thing that I do want to mention is that if we were to just go back to the stage coordinates, which are listed down here, the stage coordinates may be slightly off from the position that we return to when we come back to those stage coordinates. So there's actually some image recognition that is occurring to properly recenter our crystal for these pre-experiments. So you'll oftentimes see the sample sort of move back and forth a couple of times as you are basically matching the view that you have when you return to the crystal with the image that you have in this reference image shown here. So at this point, we go to each particle in our queue, which we could populate this with hundreds of particles if we'd like, and we'll iterate through collecting a single diffraction snapshot at each one of those crystals. After we've done our pre-experiment, something we could do is return to those crystals and set up a fully automated data collection. So what I did here first was click on my pre-experiment on the minimap, which was automatically annotated whenever I ran that queue. I've returned back to that point, and now I'm setting up a full experiment. And this works exactly the same as if I were setting up a manual data collection, except this time I'm not actually going to center my crystal and determine the tilt range. This is going to be done for me in an automated way. So all that I'm doing here is just, once again, visually identifying my crystals and adding them to a queue, which I could do based on the results of a pre-experiment if I had run that before. So after I've added my 2 grains to the queue, I'm going to go ahead and start this. And this is a point where I have sped up this process just for the purposes of viewing this. So what we're going to see is the actual automated eccentric height adjustment. So what's happening here in a sped up way is we have this crosshair in the middle representing the center of our aperture. And we have another crosshair that is basically keeping track of the crystal. As we tilt the crystal, we are bringing those 2 crosshairs together by adjusting the eccentric height. And beyond just adjusting the center of the eccentric height, we can also do occlusion detection, where we tilt the crystal to as far as we can essentially see it until it's blocked by another crystal or a grid bar. And this can be performed on both sides of the crystal. You can customize this adjustment where you sort of have trade-offs in terms of how long it takes versus the precision of the centering. After we've centered the crystal, then it will go through and do the automated data collection, and we'll even see the structure pop up here behind the minimap through AutoChem. And so this process is iterated again through everything you have in the queue. Now, after we've collected all this data, you're sort of left with potentially an overwhelming amount of data sets. And so from here, we need a really nice way to visualize those. And that can be done with the results viewer. So here I can pull up the diffraction patterns. I can look at the grains that I collected the data from. And if I had collected on multiple days, different grid preparations, I can actually take a look at those grids and see the location on the grid that I actually collected them on. If I collected a full experiment, like I did on these lower portions here, I can get an idea of what the AutoChem processing left me with in terms of unit cell and symmetry of my molecule, and I can start to sort those based on volume and unit cell parameters. I can also go ahead and quickly open particular experiments, and I can see the results from AutoChem just like I do with manual data collection. And I can open this directly in OLEX. And once again, in this case, with a fully automated data collection, I can go ahead and open up a structural solution and go from there to refine the structure. Now, one thing that is unique to electron diffraction is that we frequently need to combine multiple data sets together, and this is not always the most straightforward process in terms of knowing which data sets are best to combine together. So this is something you can also do easily from the results viewer. And what we'll see here is that we combined 4 different data sets together, and we are doing this scaling, and at the end we can take something even as low symmetry as P1 and get really high completeness data from merging those data sets together. So that comes back to that point of we don't want all of our crystals to be laying on the same orientation, because we're going to combine some of those data sets to get a fully complete solution in the end. And I don't go through too much of the structural solution and refinement part of this, because for the most part, it's going to be the same as it would be for X-ray diffraction, with just very minor differences. And what this allows you to do is when you have really large volumes of data that you're viewing in this results viewer, you can actually directly export all of those to a clustering tool. And this clustering tool allows you to get an idea of the multiple components that may be within your powder. And once you have those clusters, we've just recently added the capability to automate the merging and scaling from these different clusters so that you can feed this back into XtaLAB Pro and using AutoChem actually automate the structural solution from merged data sets as well. And so taken together, this is sort of not a comprehensive look at all the automation we've implemented within XtaLAB Pro, but I would say when you're just sort of getting an overview of the technique of 3D-ED, these are 2 really big ways that we are implementing them into this workflow. Although there's all sorts of other interesting automation features that we have implemented as well, but I just don't have time to get into all of those today. But I hope you can see that with the use of this automation, this is something that's really taking us from just using electron diffraction as a single crystal technique to really allowing us to perform these experiments where we're looking at powder materials grain by grain and allowing us to solve multiple structures from a powder mixture. And that brings me to my summary. So today we have covered the entire micro-ED workflow from sample preparation, screening, structural solution, and refinement, all within one fully integrated workflow using XtaLAB Pro and OLEX on the Synergy-ED. And I hope that I've been able to show you that electron diffraction is really developing into a broadly accessible technique that enables us to perform unique experiments with powder materials, and it can be just as straightforward as other crystallographic analyses as well. With that, I would like to conclude, and we'll take any questions that we have time for. Thank you.
Thank you, Jessica, for an enlightening presentation on the practical aspects of micro-ED data collection. We can take 1 or 2 questions now. The rest will be answered offline later. So our first question is, which holder works better for samples with solvent, a cryo holder or a liquid holder?
I would say the most common way to prepare samples with solvent would be at cryogenic temperatures. I think the case that you might want to go to a liquid cell holder would be if you're looking at a solvated sample that for some reason you're not able to look at lower temperatures. But overall, I would say that cryo is most likely the more straightforward way to prepare the samples.
Thank you. Peter asks, what criteria do you use to determine whether a particle is suitable for analysis? Is there a flow chart for that determination?
I would say typically what you're looking for is just what you would also look for in single crystal X-ray diffraction. So you're looking for something that is single crystalline, and you're looking for something that diffracts to high resolution. You can't always gauge all of this from a single diffraction snapshot, but because the data collections are so quick, you can just sort of get an assessment from that initial diffraction pattern. And if you think it's worthwhile, then it's probably not too unreasonable to just go ahead and collect a data set at that point.
Thank you. We've come to the end of our time together. If we didn't get to your question today, we'll respond offline. Note the recording will be available tomorrow. Lastly, don't forget to register for the next webinar, Episode 4, Solving Pharma's Toughest Solid Form Challenges with Electron Diffraction. Thank you, everybody, and have a good day.